Welcome to my presentation on the feeding behavior in chameleons, or the prey capture behavior in chameleons. Fascinating creatures, as we all know. Chameleons, color changing, like they're independently looking eyes, really cool stuff. But the coolest to me, at least, is their tongue that shoots out of their mouth, really elastic. It's kind of like some type of alien. It's super, super cool. But as I mentioned, we're going to be talking about the prey capture behavior in particular, and I'm going to quickly give a rundown on the steps we're going to go through throughout this presentation. So first, we're going to give a brief physiological examination / explanation of the bits and pieces that are incorporated within the experiment, or within the behavior, within the behavior. Just so y'all know all the pieces, and yeah, just helps explain the next step, which is discussing the fixed action pattern that is the behavior itself. And then through the application of prior knowledge from our lecture, from the lectures earlier, we're going to use that towards the prospect of potential research findings that can be discovered in future experiments. So, here, let's get started. First of all, we got all of this stuff is done by a paper by Ganesh Prasanna. If I'm pronouncing that right, but it's mainly done on the Indian chameleon. Super, super extensive paper, like 200 pages long. Lots of biomechanical and anatomical information on the fixed action pattern itself. So yeah, that's where a lot of this information comes from. And first of all, we're going to talk about the physiological descriptions. Quick little rundown of the physiological descriptions. We got the hyoid. The hyoid is right over here. It's a bone. It's a bone right over here. We have a hyoid just like they do, but obviously their hyoid is a lot more designed for this in particular. And attached to these hyoid muscles are the hyoglossus and the medial genioglossus. Basically, these muscles are the ones that are responsible for the push and pull, the retraction and the contraction, or the expulsion, I guess, of the tongue, basically. So you can see the muscles mapped out here. We got this and this. We got all the muscles over here, and they're mapped accordingly. And yeah, the hyoglossus is responsible for retracting the tongue, and the medial genioglossus is responsible for expelling the tongue out of the mouth. But yeah, and then that's how it does this fixed action pattern right over here. First, the first step is fixation. As you can see, the chameleon, it first spots the prey with one eye, and then it'll turn its head. And both eyes, as you can see, you can kind of see that it's facing the prey. Like both eyes are glued onto the prey, and that's once it's got its eyes locked on the prey, protrusion begins, where it opens its mouth and a little bit of its tongue sticks out. This is where it's repressing its hyoglossus in order to prepare the mouth for projection, which is when the hyoglossus stops retracting, and the genioglossus muscle causes the tongue to fire out and attach to the prey, hopefully. Interesting stuff is that it actually calculates for distance in this in particular. If it, as you can see in this diagram, if it's shot right here, then it would have missed, but so it calculates and changes the trajectory of its head so that it has it's like a drop down. It's like a drop down thing. Super cool, but also kind of tangential. Anyways, if you reach the prey or not, because it's a fixed action pattern and all that, lingual retraction will begin. Basically, the hyoglossi muscles will contract once again, or will contract this time, and after they just released to step 3, and it'll just pull the prey back into the mouth, or towards itself, and then comes hyoid retraction, which is the bone itself. You can see the bone right here, it ends right here. You can see the drop down right over here for the actual tongue, and the hyoid just retracts back into the mouth, and hopefully it got its food. It'll start chewing, if so, all that stuff. Otherwise, it'll just start the fixed action pattern again because it has the sign stimuli presented to it. It's right over here, which is the prey. That's now we got the external factors that affect the fixed action pattern. This is the quick experiment that was done. Basically, very, very simple stuff. Live and dead mealworms. They just had dead mealworms placed and live mealworms placed on construction paper, green, brown, black, and white, and they just counted how many got eaten. on this. Dead chameleons. Dead mealworms did not get eaten very much, just like once or twice, like very, very little, but it's clearly how unanimous it is that movement, because they're alive, you can see that movement is a clear indicator that triggers. So movement is clearly the sign stimuli here in this fixed action pattern, as shown by the data here, because it seems to be very correlated. Even though populations 1 in 4, like they're chameleons 1 in 4. These are just 1, 2, 3, 4 chameleons in the experiment, and they just weren't eaten here. But regardless, it's very, very clear that they eat it when it's moving and they don't when it's dead. Background didn't affect it much, as seen here, but yeah, that's pretty much it. And that's kind of all we know, to be honest. We don't really know. We know a lot about as shown by this right over here, this fantastic paper. We know a lot about the anatomical and biomechanical research. We have a lot of information on this, rather. A lot of information on the fixed action pattern in particular, but we don't really know much about the neural mechanisms that are involved. So that begs the question, how do we study prey capture responses? The fixed action pattern is clearly defined as the sign stimuli as well, as we saw earlier through this and this. But certain brain areas are surely utilized in this fixed action pattern, right? As represented in previous instinctual fixed action patterns, like the toad, as shown in the food prey and the prey response in the toad, as mentioned earlier in the lecture. Surely there must be regions in the brain that are dedicated to this, right? And that's where the experiment comes in, which is with electrodes. We can insert the insertion of electrodes into the parts of the brain. It's very, very standard. We've been seeing the usage of electrodes inside animals for pretty much the entire semester, and that's how we have all the knowledge we have. And plus, we already have knowledge of a similar circuit, as you could potentially infer. We have a similar circuit to reference, which is as I mentioned earlier, the prey response, the worm/anti-worm responses inside the toad's brain. That neural circuit seems to be very, very, very similar to the fixed action patterns that are here. Basically, movement triggers the sign stimuli, and then they do the movement, which is just extend their tongue and try and eat it. So we have a similar circuit to reference, so we can insert electrodes in the various parts of the brain and just record the action potentials that happen, is what we got. As we saw earlier in the previous worm feeding response in the toads, topographical maps in the optic tectum, and the optic tectum, as shown in this diagram right here. It's clearly a part of the brain here. We got olfactory tracts, cerebellum, all that stuff. And we can just record from all these areas, stick an electrode in here, and see what action potentials we get out of it. That's pretty much it, guys. Here are my references, and yeah, good luck on the final, y'all.